Good morning and welcome to everybody. On behalf of Lymphoma Australia and the Australasian Leukaemia and Lymphoma Group, I welcome everyone to our, what I expect will be a very highly informative virtual education session for CLL. I hope everybody is staying well and safe. And one of the unusual things that's happened because of these very strange times is that the role of virtual meetings has sort of taken on a far greater precedence, and it's fantastic to be able to touch base during these very unusual times. I'd like to particularly welcome to today's meeting our international guest speakers. We have Professor Matt Davids and also Dr. Brian Kaufman. In addition to that, we also have Deborah Sims, who will be joining myself for the patient panel a little later on, and hopefully between the two of us will provide any Australian perspectives to the meeting. So who am I? So my name's Jason Butler. I'm a haematologist in the public system at the Royal Brisbane Hospital. I'm also work with Icon Cancer Care with the blood group there as well. So just a couple of meeting points. So you can post any questions that you have, and please do that by typing them into the chat room, which you'll see at the bottom of the screen, hopefully. Donna and Sharon will be helping to moderate those questions, and we hope by the end of the meeting's agenda we'll have every single one of those questions answered. So we're going to have plenty of time for that. So the basic order of today's meeting is that we will have a presentation initially from Professor Davids, and following on from that, a talk from Dr. Kaufman. And at that point, we'll switch to the interactive panel for discussion and presentation. Now, I understand we've got a very quick poll question that we're going to bring up.
It's to the right of the video screen.
All right. I can't see that on my screen, Mike, but...
It's on the live site. Everyone, if you'd like to do the poll to the right of the video screen, please enter it into the chat in the pigeonhole screen to the right of the video, and we can move on.
All right. Excellent. Thanks, Mike. All right. So without any further ado, I'd like to introduce our first speaker. So Professor Matt Davids is an Associate Professor of Medicine and Director of Clinical Research in the Division of Lymphoma, and an Associate Director of the CLL Unit at the Dana Farber Cancer Institute in Boston. He's a leading clinical investigator in the development of the BCL2 inhibitor venetoclax, which is now available for routine use in our own country. He also pioneered the use of checkpoint blockade to treat patients with hematological malignancies who relapse after allogeneic stem cell transplants. Professor Davids has an extensive role on investigator-initiated clinical trials, particularly of novel agent combination regimens in the treatment of patients with non-Hodgkin's and CLL. So welcome, Matt, and thank you for your presentation today.
Thank you so much, Jason, and good day, everyone. It's truly a pleasure to be here. Just going to load up my slides here. So this was the one and only time that I was in Australia. This was the IWCLL meeting in 2015 in Sydney. I had a lovely time there out on the harbour, and made it down to Melbourne as well on that trip. So hopefully I'll be able to come back and visit you all in person sometime. But happy to be able to take advantage of the virtual platform here to share with you my thoughts on CLL now in 2020 and beyond. These are always challenging talks to give because I know a lot of patients come in with a mixed background in terms of their level of familiarity with CLL. Some patients may have had the disease for years and be experts in the area, and others of you may be new to the disease. So what I'll try to do is kind of cover the core and very basic material about CLL, and then a little bit about the edge and where we're headed in the research directions. These are my disclosures, including to Janssen, who's helping to sponsor this event. All right, so I just wanted to quickly put CLL in context of other lymphomas. We think of CLL like a type of non-Hodgkin lymphoma because it tends to behave similarly with involvement of lymph nodes and generally a relatively indolent disease course, at least initially. You can see that in the United States, we have about 66,000 new cases of non-Hodgkin lymphoma per year. And most of these are what's called B cell, meaning they're coming from a type of white blood cell called a B lymphocyte. And there's a mix of more indolent or slow-moving and then aggressive lymphomas, and CLL falls within the more indolent side. And it's estimated in the US that we have about 19 to 20,000 new cases of CLL or SLL per year. As you may be aware, the median age at diagnosis is about 70 to 72, and it's very common for patients to be diagnosed on routine blood work when they're feeling fine otherwise. There's a lot of variation in terms of how the disease can behave, and we've developed a number of important biological predictors based on the genetics of how patients will respond over time, and I'll review briefly what some of those key markers are. We know that patients with early stage CLL who don't have any symptoms can safely be observed, sometimes for many years. But that as patients develop more advanced stages of the disease, particularly as they have symptoms, that treatments such as chemoimmunotherapy can be helpful. And historically, these chemotherapy-based approaches have made the disease highly treatable, but historically, these are generally not curative, so the disease would typically come back over time. And in the past, we used a cellular therapy called bone marrow transplantation when you get cells from a donor. And that could potentially lead to long-term survival, but that also has some significant risks to it. So really over the last 5 to 10 years, the development of what we call novel oral agents have revolutionized the field. These tend to be oral drugs that are targeted at killing CLL cells, but tend to spare the other cells in the body. And they've proven to be both more effective and better tolerated than our traditional chemotherapy-based approaches. So just quickly, how do we make a diagnosis of CLL? So if you look on the picture at the right, you see a cell with a bunch of different proteins sticking off the surface. And we can use a diagnostic procedure called peripheral blood flow cytometry. So this just comes from a simple blood count, but it's run through a special machine that can look at what's called the ABC, or absolute B cell count. And you can see a characteristic pattern of these proteins that you see listed here. And we know that when we see those proteins on the surface of a B cell, that it's abnormal. But depending on the number of these cells, that can affect the specific diagnosis. So for example, we can see the absolute B cell count less than 5,000 with those markers. And that's a condition we call monoclonal B-cell lymphocytosis, or MBL. You can think of this as kind of a precursor to CLL, but it's not actually a malignancy in and of itself. We can also sometimes see lymph node enlargement without a lot of these cells circulating in the blood, and sometimes a lymph node biopsy can be performed that can be consistent with SLL, or small lymphocytic lymphoma. And these cells look the same as the CLL cells, but they're predominantly in the lymph nodes, and rarely will need a bone marrow biopsy to make the diagnosis. So one key point of confusion sometimes is the difference between SLL and CLL. And I'd really encourage you to think of these as part of the same disease continuum. We really think about the prognosis as being the same, and the management and treatment are the same. So if you have SLL or CLL, we really approach the disease in the same way. This is what CLL looks like under the microscope. You may not have ever seen this before. We can actually look in the blood and make what's called a smear of the blood and look and directly see the red blood cells, as you see normally here, but then lots of these little blue cells, which are CLL cells. You can see some of them, like in the middle there, are a bit smudged. That's because they're more fragile than normal lymphocyte cells. And in a field like this under the microscope, we might normally expect to see maybe 4 or 5 of these white blood cells. But here you can see dozens of these cells, and we know that this is very abnormal and consistent with CLL. There's a couple of different staging systems we can use, the RYE system and the Benet system. I'll show you the RYE system just as an example here, since we use this more frequently in the US. And you can see that patients with early stage disease can have elevated lymphocyte counts in the blood. They can have enlarged lymph nodes or enlarged spleen. But these are typically still considered the earlier stages of the disease and generally don't require treatment. Whereas patients with more advanced stage disease, as they develop anemia or low platelets, typically do require treatment. And so at diagnosis, we don't necessarily need to perform bone marrow biopsies and CAT scans because this is a clinical staging system. It doesn't require on those other tests to make the stage. So I'll go through some of the prognostic factors. One of the oldest ones is the beta-2 microglobulin, which is a simple blood test. And when that's elevated, it can suggest the CLL can behave a bit more aggressively. But much more important now are some of the more modern prognostic factors: cytogenetic abnormalities, IGHV mutation status, and what we call somatic mutations. And I'll review all of those in some detail. And a lot of people will see ZAP70 and CD38 coming up in their reports, and those still have some prognostic validity, but they don't tend to be as influential as the IGHV mutation status. So whenever possible, we do try to check that. If we don't have the IGHV mutation status available, then we can use the ZAP70 or the CD38 as a surrogate for it. So what is FISH? FISH stands for fluorescence in situ hybridization. This is a fancy way of looking at the chromosomes inside the CLL, and we can sometimes see little bits and pieces that are deleted, or we can see extra copies. So on the top here is a karyotype, a routine karyotype, where we're actually just looking to see visually if we can see any chromosomes missing. You can imagine that's not the most accurate way to examine this. And that's why the FISH was a big innovation. As you see in the lower left, that when we have 3 copies of trisomy 12, you don't have to be a pathologist to see that there's 3 red dots there and only 2 of the normal green dots. And so it's very clear that that's trisomy 12. And that really led to an improvement in our ability to prognosticate the disease because we could accurately detect the chromosomal abnormalities that were present. You can see on the right the common percentages. So deletion 13q is the most common. Over half of CLL patients will have that at diagnosis. And then you can see a variety of other markers listed there. What about the IGHV mutation status? So this stands for immunoglobulin heavy chain variable region. Don't get too bogged down in the details, but really this divides CLL patients into a couple of different categories. On the left, you see what's a somewhat more unfavorable category traditionally, where these cells are signaling more actively because they have an unmutated V gene. And this can lead to both increased cell death, apoptosis, but also increased survival and growth of the CLL cells. So these patients with unmutated IGHV tend to require treatment sooner. And traditionally with historical chemotherapy regimens, the durability of the response would be less. Although the good news is that with some of the newer treatments with novel agent-based approaches, we're seeing equivalent responses in these unmutated patients. On the right side is the IGHV mutated. These tend to be B cells that are not very activated, and they can still slowly accumulate over time, but they don't tend to behave as aggressively as the IGHV unmutated cases. And these patients also can respond very nicely to treatment. Some patients with IGHV mutated disease may actually never need treatment for their CLL. They may stay on observation for decades of time. This is a complicated slide just to show you that we've identified a number of new somatic mutations. So these are not bits of chromosomes that are missing. The chromosome looks intact, but the DNA sequence has changed a bit, and it changes the function of the proteins. And just to highlight, there's a number of different mutations we can see across pathways. If you had one mutation to take away from this morning to remember, it's on the top left. It's the TP53 gene. This is really a key molecule because it transmits the signals into the cell to die in response to therapy. And particularly when we give chemotherapy, if this TP53 gene is mutated, we can give all the chemotherapy we want, but that signal for the cell to die is not going to be transmitted, and therefore the patients are resistant to the chemotherapy. So as we transition to talking about treatment, I've had patients ask me, I have lymph nodes that are enlarged. Why can't you just cut it out, doc? And unfortunately, because CLL is truly a leukemia, it spreads throughout the body. And so, by definition, it's gone through the blood to different sites. And so we could cut out one lymph node site, but it's going to pop up somewhere else. And so we really need to think about systemic treatments that go throughout the body to effectively treat CLL. But first, we always have to decide when to treat. So these are the indications for treatment. If patients have low blood counts, and this would be due to the bone marrow getting filled up with CLL cells. Your bone marrow is like your factory for making blood cells. And so if the factory floor gets all gummed up with CLL cells, then you can't make your normal red cells, and that's what we call anemia. Or you can't make your normal platelets, which we call thrombocytopenia. And that's a reason to treat, to clear out that bone marrow space and allow your normal blood cells to be made again. The lymph nodes can become bulky or rapidly enlarging, as can the spleen. And so that could be a reason to treat. Patients can develop symptoms in the setting of these other features, and these could include fevers or night sweats, unintentional weight loss, or worsening fatigue or lymph node pain. Sometimes patients can have what's called autoimmune conditions, where the CLL can cause the body's immune system to destroy other things like other blood cells. And if those are not responding to things like steroids, then we can sometimes treat the CLL to help the problem. And then lymphocyte doubling time of less than 6 months. I say plus or minus here because it's not a hard and fast indication for treatment, but certainly as the lymphocyte count in the blood is doubling more quickly, it does catch our attention, and it causes us to watch our patients more closely for the need for treatment. So if none of the above are present, we recommend observation, or what we like to call active surveillance, because we're actively monitoring patients, but we're not necessarily treating. And why is this the case? Why do we do this? It's kind of counterintuitive. Probably a lot of you wondered when you were diagnosed, if I have leukemia, why are we not treating it? But I can reassure you that this is not just a guess. This has actually been studied quite a bit, actually, in the 70s and 80s in a variety of randomized trials where patients at diagnosis were randomized to either an early intervention strategy with chlorambucil or waiting until they had some of those symptoms and then treating at that point. On the right here, you can see in this plot where the 1 mark is, any line that crosses 1 means no difference between the immediate or deferred treatment options. And you can see across all of these different studies, these lines cross 1, meaning that there's no improvement in the survival of patients who were treated earlier. And certainly patients who were receiving these treatments earlier had longer time periods where they had side effects and other complications from treatment. Now, this has been revisited recently because people have said, well, we have these novel agents now that are better tolerated and more effective. What if we do the novel agents as an early treatment strategy? Perhaps there we can show a survival benefit. And just last summer, for the first time, we saw some early data from the CLL-12 study being run in Europe. And this is taking CLL patients with either intermediate, high, or very high-risk disease and randomizing 1-to-1 to either one of the newer drugs, ibrutinib, a pill, or a placebo, and then following these patients out over long periods of time to see if there's any benefit in terms of the survival for the patients who were treated earlier. This is what we call an event-free survival curve. It's a bit complicated here, but I'll walk you through it. So at the bottom on the x-axis here is the time until an event. An event could be that the CLL worsens to the point where it needs treatment, or the patients have a significant side effect that they have to stop the treatment, or if the patient died for some reason, that would count as well. And then on the top is, on the y-axis is what we call event-free survival. And in this graph here, the blue curve is the patients treated with ibrutinib, and the orange curve is patients treated with placebo. So at first glance, it may look like there's a significant advantage to the ibrutinib because the event-free survival is better with ibrutinib than with the placebo. But actually, if you look at the reasons why the patients on the placebo had events, in nearly all cases, the reason was that they had progression of their CLL, which then required treatment with drugs like ibrutinib. And if you look at the overall survival of the patients, there's no difference between the two arms. So even though taking the ibrutinib prolonged the time until another treatment was needed, it's possible that treatment wasn't even needed in the first place. And we certainly did see some side effects in the patients treated with ibrutinib. And so clearly right now, there's no advantage to an early intervention with ibrutinib if it's not going to make patients live any longer and they're going to experience the potential for side effects. So often then patients ask, well, then what can I do to slow this down? Are there any nutritional interventions, complementary medicine, et cetera? Unfortunately, this is not an area that has a lot of data, but I will show you one slide of data from the Mayo Clinic that used a green tea extract called polyphenol E. This is very, very high dose green tea extract. This is not something you could get by having a couple of cups of green tea per day. You can see at the top, this is every bar represents a patient, and the percentages here represent a reduction in an absolute lymphocyte count in the blood. And at the bottom, a similar type of graph, but looking at the lymph node reduction. So certainly there was some biological activity of polyphenol E here. There was a reduction in the disease burden. But we've never seen that this intervention has actually prolonged the time until a true CLL treatment is needed, and certainly no data to support this prolonged survival of patients. And at this high dose, again, there are some risks and side effects. There's risks of liver injury and so forth, and gastrointestinal side effects. So this is not something I routinely recommend for my patients, but it is certainly something that could be considered. So what about the more robust CLL treatments? So going back to the 1960s, we had drugs like chlorambucil, which could palliate the disease and certainly was helpful from a symptom management standpoint. But the rates of complete remission were very low, only about 5%. As we moved into the 70s and 80s, more powerful drugs like fludarabine came along and the complete remission rates began to go up. In the 90s, we began to add 2 different chemotherapy regimens together and achieved complete remission rates close to 30%. And you can see that by sort of the early to mid 2000s, that very powerful chemoimmunotherapy regimens had been developed. Regimens you've heard of now probably called FCR or BR. And you can see the complete response rates would go up as high as 70% in some of these studies. However, as we discussed earlier, chemoimmunotherapy has a fair amount of toxicity. It can't be easily reused multiple times. And so that's what really inspired the development of the novel agents in the 2010s. And you can see them listed here. The CR rates are variable, but the efficacy is pretty uniform. These are very effective drugs. So this is the data for time-limited FCR chemoimmunotherapy, and there's multiple studies showing the same thing, that there is still this potential for long-term benefit for younger fit patients with mutated IGHV. You can see in the green box there that outwards of 6 to 7 years after starting therapy, these patients who got 6 months of FCR, about half the time or more, are still in remission, suggesting potentially even functional cure with this regimen. However, for patients with the unmutated IGHV, these other dotted lines, you can see that they do tend to steadily progress over time. So as I said, there's quite a few new novel agents. We have them all available to us, fortunately, here in the US now, starting with the obinutuzumab regimens, We have ibrutinib, we have PI3 kinase inhibitors like idelalisib and duvelisib, venetoclax, and then most recently acalabrutinib. So if you start to imagine all the different unique sequences and order of administration of doing these drugs, it actually winds up of 720 unique sequences that you could get as a way to administer these drugs. So with all of these new options, new challenges have emerged on how best to sequence these therapies and how to manage patients who progress after all of these drugs. So one approach that we've studied at our center is rather than abandoning chemoimmunotherapy completely to try to add to it. So we recently published our study of ibrutinib in combination with FCR. And in this study, we showed the best response rate in terms of the bone marrow MRD that's ever been reported for a CLL regimen, 84%. MRD means minimal residual disease. So we look at in patients where we can't see any CLL under the microscope, and then we can use very sensitive molecular techniques to see if we can even detect any CLL cells in a molecular sense. And in our patients, 84% had undetectable MRD, and that was irrespective of their IGHV mutation status. So we don't have long-term follow-up yet on this approach, but we think it is one of the promising ones for the younger and fitter patients. But most CLL patients are older and have other medical issues and probably are not good candidates for chemoimmunotherapy. So what is the optimal novel agent-only approach for frontline CLL? And there's a couple of main ways that this is developed. There's a way of giving continuous therapy that can be highly effective. And there's a newer way that we can give time-limited therapy for a period of time and then stop, and patients can enjoy long remissions. So in terms of the continuous therapy approach, we have the longest term data with ibrutinib. This is an oral agent taken once a day. And you can see on the left side with this bar graph in terms of the overall response rate, even by 6 months, over 90% of patients have responded to ibrutinib, meaning their CLL lymph nodes have shrunk down, their blood counts are starting to improve, they're feeling better. You can see that over time, as you look at the rates of complete remission in the darker green at the top, that close to 30% of patients will begin to develop complete remissions as you get 4 to 5 years out from starting. And this translates into a very nice progression-free survival. About 75% or so of the patients have not had progression of the CLL after 5 years on ibrutinib therapy, certainly far better than the older therapy chlorambucil that we used to use in the past. There's another BTK inhibitor drug, so similar mechanism as ibrutinib. It's called acalabrutinib, and this is also now approved in the US. You can see the progression-free survival curve from one of the early studies with acalabrutinib also looks very good, with very few patients progressing after 4 to 5 years of therapy, and very high response rates. So this is another very effective option for CLL patients. In the US, we have a large group called the Alliance Group. It's many different centers that come together to run these clinical trials. And in this trial, they looked at whether adding rituximab, which is an infusional antibody, to ibrutinib was helpful. And it turns out that ibrutinib on its own did just as well as ibrutinib with rituximab. So we usually just use ibrutinib as a single drug. But interestingly, they did a similar comparison with acalabrutinib and a different antibody. So similar class as rituximab, but this antibody is called obinutuzumab. It's a little more potent than rituximab. And you can see these top two lines here. The top one is the doublet of acalabrutinib with obinutuzumab. And it does look perhaps a little bit better than the acalabrutinib on its own. Now, the statistics of this are not completely clear, and so this is not a very clear benefit, but it's more of a hint of a potential benefit. And it was enough to allow our FDA in the US to approve both regimens for our CLL patients, and we can discuss with them the pros and cons. An advantage of the antibody infusion is it may prolong the progression-free survival, but there's the inconvenience of getting the infusions and some additional risks of toxicities. But what about if there were a way to have a highly effective but time-limited novel agent-only regimen that could be applicable to a diverse array of CLL patients? In other words, could we have our cake and eat it too? Could we have the benefits of the novel agent approach, but also the benefit of the time-limited approach like we used to have with chemotherapy? And that's the principle behind this regimen of venetoclax with obinutuzumab. And this was compared to chlorambucil with obinutuzumab, and certainly the venetoclax-based regimen was superior. If you look out 36 months from starting treatment, 82% were still in remission compared to only 50% with the chlorambucil regimen. But what I like to do on this curve here is to look at the 12-month mark and remind people that after 12 months, that's when people ceased therapy. So they only got 12 months of treatment with the venetoclax. And you can see that top black line, although there are still some patients having progression of the CLL, overall patients are doing quite well, even for 2 years plus off of therapy. So as we think about how to treat frontline CLL, meaning for patients who've never had treatment before, we have these two great options. We have the BTK inhibitors, and then we have venetoclax and obinutuzumab. So advantages of the BTK inhibitors, we do have the longest term data. We have some patients on ibrutinib now, 8 or 9 years, still doing very well. Very convenient, just pills. There's no infusion. There's no need to monitor for other laboratory abnormalities, like what we call tumor lysis syndrome, when tumor cells are bursting open quickly, as we can sometimes see with venetoclax. We have our highest level of evidence, what we call phase 3 data, compared to our best chemotherapy regimens. And we have a lot of data now accumulated for the fact that venetoclax can be a very effective drug after ibrutinib in patients who have progression. Now, on the flip side, we have the potential with venetoclax obinutuzumab for a 1-year time-limited therapy, which a lot of patients find appealing, the idea they don't need to stay on long-term therapy. There's no known cardiovascular or bleeding issues with this regimen, as we can see with the BTK inhibitors. There's less concern that we need to remind patients, and patients need to be reminded to stay on their long-term therapies. And potentially there could be cost savings for society if 1 year of therapy is very durable, because the BTK inhibitor drugs are very expensive to give year after year. We're trying to put 3 of these drugs together, acalabrutinib, venetoclax, and obinutuzumab, in an ongoing study called AVO at our center. And the idea here is that even though we're using 3 drugs, it's a bit complicated, that we could try to do only about 15 months of therapy and have an even more durable response than they might be seeing with the 1 year of venetoclax obinutuzumab. And so far, our data look very good, but it's early on. So in the last few minutes, we'll just talk briefly about relapsed refractory CLL. In other words, patients who have already had some initial treatment, they've gone into remission, and then the disease has come back, or the rare patient who received initial treatment and did not have a good response to it. So a regimen that has been developed around the world, but one of the leading sites was in Melbourne at the Peter Mac. John Seymour presented these data and has been published now in the New England Journal, comparing venetoclax with rituximab to a standard chemotherapy course with bendamustine and rituximab. And you can see very nice results for the venetoclax regimen, again with an advantage being that this is a time-limited regimen. And these patients got, in this case, 2 years of therapy. And 2 years later, after 4 years after starting, more than half of patients are still in remission. And of the patients who made it through the full 2 years and received all 2 years of treatment, 68% are still progression-free 2 years later. What about combining ibrutinib with venetoclax? That's the so-called IV regimen. This is also a promising new combination for relapsed refractory CLL. There's been a couple of really good studies around the world, one in the UK and one in the US here at MD Anderson. Remember that MRD test I mentioned, minimal residual disease? So this is looking even at a molecular level. You can see 54 and even 68% of patients by 2 years have undetectable MRD in their bone marrow. So very deep remissions, and this is an all-oral regimen, which is very convenient. We've taken another approach of trying to combine ibrutinib with another drug that's in development called umbralisib that targets the PI3 kinase. This is another all-oral regimen, and we saw high rates of complete remission, close to 30%, and good durable responses. But this type of regimen does require continuous therapy. And then recently we saw the first randomized study comparing 2 different novel agent-based approaches in CLL. This study compared acalabrutinib, which is the top line here, to idelalisib, a PI3 kinase inhibitor, or bendamustine rituximab chemotherapy. And you can see that the acalabrutinib was superior to both of those regimens, making this another good option to consider for patients with relapsed CLL. The other thing I'd like to highlight is that there's a whole new generation of BTK inhibitor drugs that are on the horizon. One of the issues with the current BTK inhibitor drugs is that there's resistance mutations that can be described that have occurred in the BTK gene that lead to the protein not responding to the drug. And the data I'm showing you both on the left and the right are from 2 different drugs. On the left is LOXO 305, and on the right is ARQ 531. These are both drugs in early phase clinical trials, but they're showing responses in CLL patients even if they have this BTK mutation. So it really provides good hope that even if you develop a resistance mutation on a drug like ibrutinib, that you may be able to continue on a BTK inhibitor drug from this new generation. One of the feared complications in CLL is Richter syndrome, which is an aggressive transformation of CLL into more of a high-grade lymphoma. There's a number of promising studies going on to treat this condition around the world. I just wanted to highlight our work here, combining venetoclax with chemotherapy, our EPOC, and we've seen very high rates of response. We've seen complete remission rates over 50% in this regimen, whereas with chemotherapy alone, it's usually more like 15 to 20%. So we think this is a promising approach even for the highest risk patients who develop Richter syndrome. Finally, I just wanted to touch briefly on CAR T cell, chimeric antigen receptor T cells. These are the super soldiers of the cellular immunotherapy world. The idea here is to take T cells from CLL patients to actually introduce using a retroviral vector, something called a CAR, chimeric antigen receptor. You can see on the forehead of this CAR T cell. And these are proteins that actually can selectively recognize CLL cells. So these CAR T cells can then be reinfused back into the patient, and they can go around and act as super soldiers, killing off CLL cells. I think Brian may touch briefly on this as well. This is an incredible treatment that actually has curative potential for CLL patients, even those patients who have already had all of the other drugs. And these are some of the promising studies that have been done on CAR T cells. You can see high rates of overall response in the sort of 50 to 90% range, maybe 40 to 45% of patients in some of these studies achieving complete remission. So very impressive results. This is one of the studies that was recently presented and again, showing 45% rate of CR. So kind of putting things together, we've certainly had an evolution, I would argue even a revolution in CLL therapy over the last couple of decades. We started with chemotherapy, which could certainly get you from point A to point B in many cases, but had its limitations. Chemoimmunotherapy was like a motorized car, certainly a dramatic improvement over the horse-drawn carriage with chemotherapy, but again, with its significant limitations. Now we've had the agents coming through as novel agent monotherapy and various combinations, but novel agent monotherapy, even on its own, is fantastic. Patients can do well on drugs like ibrutinib for many years. This is a huge advance. But now as we get into this era of novel agent combination therapies and CAR T cells, I mean, we're really seeing incredible results. I think there's lots of reasons to be optimistic. Maybe we're not quite at flying cars yet, but we're getting pretty close in terms of these great options for our patients. So last slide, just to kind of sum up here, I would say that we've kind of reached the end of the beginning of what we call the novel agent era. And we now have this powerful toolkit of novel agents with many more coming along. So you have this idea of sequential novel agent monotherapy, and that may be best for some patients. So it may be best to start with ibrutinib for a few years and then go to venetoclax and then go to a PI3 kinase inhibitor. But for some patients, time-limited combination approaches may be better. I think particularly younger patients and those with higher risk disease may benefit most from this approach. But I would say also that optimal sequencing and combination of these agents has yet to be defined. There's so many different combinations to explore and a number of important clinical trials that are ongoing. So for the patients out there, I'd certainly encourage you to consider active enrollment in clinical trials. This really remains critical to the mission of optimizing our care for CLL patients. And with that, I will conclude. Thank you.
Hello, hi, Dr. Brian Kaufman here. And let me just share my screen with you.
Yeah, thank you, Doctor. If you could continue on.
Thanks to Dr. Matt Davids and thanks to Lymphoma Australia. I had the pleasure of being at that same IWCLL meeting that Dr. Davids referenced. And we actually did a patient educational session that was really well attended in conjunction with that. And got to meet some of the fine doctors and patients that were in Australia. And I'm looking forward to getting back there again for another visit, but happy to be here virtually now. So I'm Dr. Brian Kaufman. I'm a family doctor. I'm not a hematologist or oncologist like Dr. Butler or Dr. Davids, but I'm a retired family doc. But I'm also a CLL patient. I'm the co-founder and executive vice president, chief medical officer of the nonprofit CLL Society. And we're lucky to be able to work with people like Lymphoma Australia. I always tell patients it's a blessing because CLL is both a leukemia and lymphoma. So we benefit from all the research and benefits that are out there for both in the leukemia world and in the lymphoma world. My learning objectives for today is I'm going to be talking about how to be your own best advocate. I'm going to use a program that we have, Test Before Treat, and Dr. Davids did the heavy lifting for me on this, so it'll make my job a lot easier. I'm hoping that by doing this together, we can increase our odds of staying alive with CLL and I'm going to talk a little bit about shared decision-making to improve outcomes. So this is my story. I was diagnosed 15 years ago at the age of 54, so you can do the math and figure out how old I am. And as Dr. Davids talked about in these terms of these prognostic markers, if it was bad, I've had almost all of them. Yet I got 7 years out of a phase 1 first large trials of CLL. My video okay there?
Thank you. Got it now.
Yeah. I got 7 years out of a drug that didn't even have a name, PCI 32765, just a number that later became ibrutinib. I had a CAR T therapy, which was difficult, but when I was restaged later at 1 month, now you know what undetectable MRD means. At 1 month, 13 months, and 23 months, there was no CLL to be found, less than 1 in a million CLL cells in my bone marrow. So I started to tell my story about what was going on there, and I started with a blog that maybe some of you have seen. Just take a glance in the mirror here, because I think as a physician, I was able to see around the corners and see some things that were maybe other people didn't get to see, and wanted to share that. Because I didn't think it was fair. Being a doctor, I had certain advantages, and I wanted to make sure other people's had that advantages. So I think you'll find I'm one of the most empathetic doctors around. And we established the nonprofit CLL Society to try to get the word out about CLL. And this is our website here, and it's got lots of information for you to learn about. And all our services are free. Our motto is smart patients get smart care. I'm going to talk about why that's important. We have a toolkit there, and all of these are downloadable where you can find out. There's a lot of vocabulary for people that are new to this. There's a lot of acronyms and words that you'll hear for the first time. It can be overwhelming, and we try to help with that, but it's an iterative process, and the more you come back and the more you learn and the more you attend things like this a webinar being put on by Lymphoma Australia, the more comfortable you get with these languages. These are some of the programs we have, support groups and educational forums. We work a lot with Dr. Davids and others. And I'm going to spend a little bit of time on the testing issues, both at diagnosis, testing before treatment. We covered UMRD. So I'm going to now talk a little bit about how to be your best advocate and how to save your own life and maybe save the lives of others. And what I'm going to say is that there's no space between being your own advocate and advocating for others. When you're helping yourself, especially if you're telling your story, you're helping others. And when you're helping others, you are helping yourself. And if you can keep that in mind, it really helps with the journey. And I'd be interested in Deb's reflection on that when she talks about this, but that's certainly been my experience. So I talk about being a smart patient, and the first step in that is knowing yourself. You need to know a little bit about your cancer. That's why we say smart patients get smart care. But you also have to know if you have other medical issues going on. Are you diabetic? You have hypertension? You have irregular heartbeats? This is going to affect what therapies you have. Do you have bleeding problems? Are you on a blood thinner? All of these things will make a difference. Are you taking medicines for peptic ulcer disease? Some of the medicines for that can affect, you have to know all of these things and be aware of these things. You also have to know what you're interested in. Now, with so many options, we have an embarrassment of riches in terms of treatments. What kind of treatment do you want? Do you want something where it's 6 months and done and you don't care that it's chemo? Do you want a limited duration therapy? You don't mind taking a medication long-term if it's a simple pill that's very effective? You have to know what your own bottom line is, what your own preferences are. These have to help guide you, and nobody's going to know those except you. But you also have to know what's going on around you. You have to know that a lot of these agents that Dr. Davids talked about aren't always available. They might, or they may only be available at certain research centers, at certain academic centers, or there may only be a comfort level there. So if you want access to clinical trials and stuff, sometimes you have to travel. And in Australia, that geography can be really a challenge. You have to know what your healthcare team's strengths and weaknesses are. We emphasize three things at the CLL Society. One is novel therapies. Generally, we push people to consider novel therapies. 2, clinical trials, because we think we get great care there. And 3, expert care, having an expert as part of your team. We're very big on trying to get those things growing. So you have to know what's available in your community. And sometimes nowadays within the COVID environment has actually opened up the possibility of having remote consultations with CLL experts from across the country. You have to know what support you have. We talked a little bit about CAR T therapy. CAR T therapy is not a solo process. You need family or friends to support you. I couldn't have done it without my wife's help. And in every country, you have to know what your insurance is going to pay for, what's paid for by a clinical trial, what outside costs you are. You have to know what these external challenges and opportunities you have and what's available to you. So I built up some commandments that I use to try to help me navigate through the CLL. And the first is to get an expert on your team. And I've talked about why that's so important. There's actual studies that show that there's a survival advantage to having an expert. And the others are just sensible if you think about it. And Dr. Davids talked about this watch and wait or active surveillance. We have time in CLL, but we don't have forever, and you have to make some decisions. I advise people to be very zen and underreact to CLL. Almost for all of us, it's slow growing. You're going to get good news. You're going to get bad news, kind of underreact. There's more to your life than the CLL. You need to expect the unexpected and don't expect to control the disease, and understand that even not doing anything carries risks with it if the disease progresses. The most important commandment, I say, is you need to make difficult decisions with imperfect knowledge and contradictory advice. And I'm sure that Dr. Davids and Deb will echo this, because there are so many good options that are available now that it's sometimes very hard. And Dr. Davids would love to be able to give you 10-year data on this treatment or that, but we don't have it. So we have this imperfect knowledge, and different centers will present different things. You have to kind of think outside the box. You have to get local support. Money can be an issue, but also remember to live your life and to give forward. And remember that there's a lot more to life than CLL. So how to be your best advocate. I'm going to talk about appropriate testing, and we're going to focus on the one in red here, test before treat. And there's the 3 things that Dr. Davids covered: the FISH testing, the IGVH, which is also IGHV. You can see it both ways, and the TP53. These are the 3 critical tests that we recommend. And I'll see if this video is going to play for us here. And this is Dr. Anthony Mato, who's done a lot of research on CLL in combination with Dr. Davids.
Anthony Mato from Memorial Sloan Kettering, director of the CLL program, here to support the CLL Society's program, Test Before Treat. It is incredibly important in 2018 to have appropriate prognostic testing performed prior to first therapy for patients with CLL. Researchers have worked really hard to develop therapies that are targeted, and those targeted agents work well in certain patient populations. Right now, we're able to look at the molecular and genetic profile of patients and identify which patients would benefit from combinations like chemotherapy, which patients need a targeted agent by itself, and maybe in the near future, which patients should be treated with combinations of targeted agents. The data that provide clinicians the information needed to make that important choice is the data that's obtained from molecular and genetic prognostic testing, FISH, cytogenetics, array, next generation sequencing. I'm here to support the campaign because I feel that in order for a clinician to make that decision, that information needs to be considered standard of care. Those recommendations are supported across the country from organizations like NCCN, IWCLL, the CLL Society through this new campaign. So I'm hoping that some of the research that we're doing has demonstrated that that's not always done in practice, but can help to inform practitioners that that should be the way of the future for patients with CLL. Thank you.
And I would argue that that's true wherever you live, whether it's in rural Australia or New York City. You need to have access to these kinds of test before treat information. And I think, and I would push Australian patients to ask their doctors about them, inform them of them. If the community hematologists don't know, empower yourself to know what's going on here. So what is self-advocacy in this regard? This is from the Institute of Medicine. Self-advocacy refers to being assertive, not obnoxious. There's a fine line, but assertive in order to communicate clearly any needs related to your cancer care. And as a physician, let me tell you what we want from a patient. We want you to be direct. We want you to present the facts, state your point clearly, be brief. The one thing that doctors don't have enough time of is time. So if you can get to your critical points first and provide facts over opinions, we're going to deeply appreciate it, and you're going to appreciate it. I also recommend that you bring a tape recorder into your meetings with you and record the answers to the doctors, because my wife and I have often walked out of a doctor's visit disagreeing on what was said. So recording is a good way to go. But be direct, be brief, and be rational in what you're asking for. Shared decision-making, this has changed. In the old days, the doctor brought in all the medical information, but nowadays, with organizations like Lymphoma Australia, with the CLL Society, with many organizations that are out there, patients gather a ton of information, and especially for rare cancer, sometimes they can be even better informed than a community hematologist, and they rarely see CLL. So patients bring in information and need to share this with their hematologist oncologist. We bring in, as I talked before, our own needs, values, and preferences, which we've thought about before, and we mix those together and come together with a care plan and follow-up. That's what a shared decision is about. So those were my learning objectives: to learn some practical tips on self-advocacy, to talk about testing before treatment as a advocacy example, to give you some things that I found, my commandments, to increase our chances of staying alive, and to employ shared medical decision-making to improve outcomes. So I want to thank Lymphoma Australia for this opportunity to give a talk. And here's a beach in Southern California that is maybe 25, 30 miles away from where I live, just Newport Beach. So it's sweltering hot inland in California right now, and I imagine the weather's quite different in Australia. And with that, I'll say thank you and goodbye.
All right, thank you very much, Brian. I do apologise, I didn't get a chance to introduce you because my computer went into shutdown mode just as Matt's talk finished, and thank you to Matt for what I thought was also a fantastic overview of CLL. I just thought before we move on to the next section, we've got a little bit of time, I just wanted to clarify the availability of some of the treatments that Matt mentioned and Brian mentioned in Australia. So pretty much that list that he had on the slide is sort of very much in keeping with what's available here, except for duvelisib. Ibrutinib and venetoclax are available and they're funded on the PBS, but only in the relapse setting, so they're not available at this point in the frontline setting. And acalabrutinib is approved, so it's licensed to be used in Australia. It's not yet reimbursed on the PBS, but if everybody just watches this space, we'll hopefully see some very good news in the next month or so. And on the testing of the three important things, so FISH, TP53, and IGVH, they're all available certainly through the public sector, fairly easy for doctors to order. A little bit more troublesome in the private sector, where in fact each of those at this point will incur an out-of-pocket expense, and probably about $1,000 Australian to have all three of those tests done at the moment privately. We believe that the FISH testing for 17P will be available and funded sometime in the next month or two as well, so that at least will be one less expense. So moving on to the next part of the talk, which is going to be the panel discussion. And at this point in time, I'd like to introduce Deborah Sims, who I suspect many in the audience will know fairly well. So Deb is a lady with CLL, 3 children, as well as being a tireless advocate for patients with CLL since her own diagnosis back in 2011. Deb's had a very instrumental role to play in trying to gain access to life-saving treatments in this country, and continues to fight for others to ensure that there's equity of access across the board for all patients with CLL. So, Deb, do you want to sort of say a few words to start with before we move on to the panel?
Yeah, thank you, Jason. And I don't want to take too much time because this is such an opportunity for patients here to talk and ask questions of Matthew and Brian. And thank you both for your presentations. Matthew, I've been living with this disease nearly 10 years, and there were slides there that I hadn't seen, having covered lots of hematology conferences. Fantastic presentation. And Brian, thank you for all the work you do with the CLL Society. And in fact, Brian and I go way back because I was diagnosed in 2011, age 38, DEL6Q. So on that slide, I haven't seen that before. I'm one of 6% of CLL patients. And, it's negligible how many of us are diagnosed under 50 and how many of us are female under 50. So I hit the lottery. I've been buying lottery tickets ever since. I was found to have quite aggressive disease. I was SLL presentation, so lymphadenopathy, much more, swollen glands everywhere. A year of watch and wait. FCR in 2013, relapsed 6 months later after FCR, and was headed for an allogeneic stem cell transplant, which was standard therapy. I was under 40. It was a good option. Didn't have a great match, didn't have a sibling donor. And I was seeing the stats coming out on 5-year survival. And for me, it's that risk assessment, wanting to be here. My children were only 7, 5 and 2 when I was diagnosed, and I just wanted to buy myself as much time in the hope that I would see them become adults. So I went to America and bumped into Brian Kaufman, who had organised a conference for the CLL Society, and it was at that conference that I bumped into John Gribben, who told me about a trial of a drug called ABT199. And obinutuzumab, and that actually was on Matthew's slide there, which showed that obinutuzumab and now venetoclax. I was on that from 2015. So this is the important thing about being on clinical trials, because it's often the only way we're going to get access to these medicines years in advance. And at the moment, acalabrutinib, for instance, isn't on the PBS, but it is in a clinical trial. So always ask your doctors. We're very lucky here that we've got some excellent specialists, and I'm a patient at the Peter Mac. And just to bring you up to speed with where I'm at, I had no detectable disease on venetoclax for 3 years, nothing in the bone marrow. But in 2018, at the point that I stopped commuting to England to get it, because I had to enter an overseas trial, as many of you know, I started developing resistance. And I got another year out of venetoclax. December 2019, I had to come off it, and started getting quite bulky again. Went on to ibrutinib, which has controlled the disease, but I'm delighted to say that I'm now on a phase 1 trial for CAR-T. My cells were harvested 2 weeks ago. I thought I was going to have to go to America for this treatment, but I am on a trial at the Peter Mac, and I will have chemotherapy in about 2 weeks' time, and then go through the difficult journey that I know Brian went through. It's not a treatment for everyone, but I am younger, and it is a treatment that I wanted to do before transplant. So I will keep you all up to date. I will be, fighting for this treatment to become available to all of us. But I have to say, if you're relapsed, if you've been on ibrutinib for 6 months and it's your second or third line treatment, I would seriously look at this trial and talk to your doctors. So that's bringing me up to date, and back to you, Jason, to involve everyone and get some great questions out of these guys.
All right, thanks, Deb. So I think I'm going to hand over to Donna, who's going to sort of coordinate the next part of it. So Donna is the lead of our nurse program with Lymphoma Australia. So we're doing amazing jobs with education, both of other nurses across the country, as well as our patients and doctors for that matter. So Donna, I see there's a few questions on there for the panel.
Hi, thanks, Jason. Thank you, presenters. They were very good presentations and very good overview of what's going on in a very quickly changing world. We do have a few questions on the the screen. And thank you for those who have been participating in the polls throughout, and we'll be putting those results up throughout the panel discussion. So I've just got a question here that it'd probably be a good one to start off with. We've been talking a lot about ibrutinib and BTK inhibitors, and the question here from John is, how long can you stay on ibrutinib for, and how quickly can you become resistant to it? That's quite a question that a lot of patients do ask.
I can take that one.
Thank you.
Yeah. So the longest standing data that we have on ibrutinib is patients now sort of in the 8 to 9 year range after starting. Still many patients from that original study doing very well. That's for what we call treatment naive patients, so patients who are getting ibrutinib as their first therapy. If we look at patients who've had prior chemoimmunotherapy and then go on ibrutinib, the range there on average is about 5 or 6 years of remission. But again, some patients enjoying longer remissions than that, and other patients, particularly those who have that deletion 17p or TP53 mutation, a bit shorter than that. One of the things that's nice about ibrutinib is that we've seen now, in terms of the safety data, that there's no new side effects that seem to be popping up late. We do have to watch for high blood pressure in patients on ibrutinib, so we have to monitor for that. But otherwise, the toxicities and side effects we tend to see usually happen early in the first year or so. And if patients kind of make it through that time period and are tolerating it well, they often tolerate ibrutinib well for many years.
And I would just add a couple things to that, when Dr. Davids or any doctor talk about these statistics, that's predicting for a group, and individuals will have mileage will vary. I went 7+ years with 17 deletion, all those high risk, having failed a bone marrow transplant. I mean, really bad disease, and got 7.5 years out of ibrutinib before I needed to move on to CAR T. So everybody varies. And generally, if patients can make it through that first year of ibrutinib, they tend to do well. The adverse events seem actually to calm down generally. There's fewer infections after the first year. But the main reason people go off ibrutinib at the beginning is side effects, not disease progression, because people, as good as it is, there are side effects. And that's why we have second generation BTK inhibitors coming, where people can switch over, not if they've progressed on the ibrutinib, but if they've had side effects. A different drug may have different side effects or fewer side effects. So it's a really good option for patients sometimes to be able to switch. And that's why you've got to be lobbying and pushing to get these other drugs approved in Australia.
Yeah, and hopefully we'll get there very soon. What is the, so talking about acalabrutinib and some of these newer generations, are they more likely to be your first line these days in the States?
So right now it's a discussion with my patients. I do discuss both ibrutinib and acalabrutinib in the frontline setting. I think that particularly for patients who may have pre-existing cardiovascular issues, but they want to be on a BTK inhibitor drug, that's where I tend to lean toward acalabrutinib. On the other hand, if I have a patient with high-risk disease with TP53 mutation, I think we have a little more data still for ibrutinib there, so I tend to favor it there. But I think either is an option. Sometimes there's certain little things that can make a difference. So a class of drugs called proton pump inhibitors. People often use these for heartburn symptoms. If you really need that, then acalabrutinib may not be a great option for you because there's a drug interaction with that type of drug. The other thing is that acalabrutinib is twice a day and ibrutinib is once a day. So some people prefer a once a day drug. So there's differences between the two, but they're both great drugs.
Excellent.
Can I just say as well, again, it comes back to sequencing of drugs. So when I failed venetoclax at the end of last year, there was a choice between putting me on a trial of acalabrutinib or going on to ibrutinib. But my doctors, who are specialists, knew that there was a CAR T trial coming where you had to have been on ibrutinib for 6 months. So this is why it's really important to make sure you're, particularly if you're interested in joining clinical trials, that you're seeing a specialist who, or your doctor is conferring with a specialist centre where they have clinical trials, because you need, it is all about sequencing. I know people who haven't relapsed on venetoclax, but the stats of someone on a BTK inhibitor, such as ibrutinib, after failing venetoclax, who hasn't had it before, We don't know because it's not standard for someone to go onto venetoclax before ibrutinib. So, you know, the latest data out of that is only a couple of years. We know how long ibrutinib would last. So it's that sequencing that makes actually decision-making really difficult. And that's what Brian was talking about. It's a constant risk assessment. Ibrutinib might work for me for the next 3 or 4 years. We don't know. So should I jump now into CAR-T when my bloods are as good as they've ever been? You know, and that's a dangerous thing to do. So it's that conversation constantly with your doctors about what the outcome is that you want. Is it keeping the disease quiet with ibrutinib and having a healthy life? Is it getting rid of the disease altogether, but there may be some risk in going for that? We're very lucky. We've got great doctors here. So just ask about sequencing of treatment and clinical trials. If I'd gone onto a acalabrutinib, I wouldn't qualify now for this CAR-T trial. And that this is a once in a life opportunity for me to have this, because the chances of us having CAR-T in Australia for CLL is amazing. As Brian said, I mean, even centres in America, there's not many trials for CLL. So we're very lucky to have this. And it's still recruiting people.
And I would add to what Deb says, this is about knowing yourself. I mean, some people would say, I'm on the ibrutinib, it's working. Let me kick this can down the road as far as I can. Let me stay on it as long as I can. And others want it over with. Well, they said, I've been really tough. When my disease goes south, it goes south fast. So while it's under control, I want to get into the CAR-T trial because that has the potential to be curative. I mean, there are studies out of the National Institutes of Health that people are more than 8 years out with no evidence of disease, more than a decade out. It's just a handful of patients, but it certainly gives us some hope. And the only curative therapies for certain that we know in CLL are cellular therapies like transplants, and this is a cellular therapy. So I'm not saying it's curative, but I'm saying there's a hint, there's a chance for some people it might be. So, you know, it depends on what direction you want to go and what's important to you in making these decisions. You have to share that. And I'm going to add one other nuanced thing in terms of this, making these decisions. If you want to get to CAR-T, you have to have a way to get to it. And if your disease is raging out of control, you have to find a way to get your disease under some control, because the CAR-T can't deal with disease when it's spreading like wildfire. It needs to be somewhat controlled for the CAR-T cells to work well. So you have to think about CAR-T not as your last therapy, but kind of as your penultimate therapy. How am I going to get to it? I just wonder if Dr. Davids has any thoughts on that particular issue about bridging to CAR-T.
So I do agree with what you're saying to some extent, although I want to draw a contrast a little bit with allogeneic stem cell transplantation, because I do think it's a bit different there. So It's very clear with allo transplant that the deeper the remission you're in when you go into transplant, the better the outcomes are. So if you're in a PR that's not as good as a CR, in other words, partial remission or complete remission, if you're undetectable for MRD, you have even better outcomes going into allo transplant. I don't think we know quite as much with CAR-T yet. We've seen patients in a PR go into CAR-T and have fantastic responses. So I agree with you. You don't want to go in with your disease growing out of control, but there may be a little bit more leeway there with CAR-T as compared with the allo transplant.
And just in terms of, again, sequencing, CAR-T, there's a debate about whether it should be before or after transplant. But here, you wouldn't be able to get CAR-T if you'd had an allogeneic transplant. So I've made the decision to go the other way. But in the States, you can. Is that right, Brian?
My understanding is that first, CAR-T is only experimental in CLL, but a number of the trials that are open will allow people who've had an allogeneic stem cell transplant. They allowed me, and I'd had a transplant. So I think most trials are allowing people because, and that's often the way it is at the beginning of, it's the sort of the worst of the worst, the people who failed everything get the trial drugs, but then later on, they get more moved front line and are used with people who've had less therapy.
And I would say, and this is not at all to minimize the risks of CAR-T, because it can be quite severe, but I would say comparing across allo transplant risks to CAR-T, the risks of transplant are still higher. So I do think, I certainly agree with Deb's approach. If you have access to a CAR T trial and you can do that first, it makes sense, and save allotransplant for down the road.
Excellent. Look, I had a question here back to BTK inhibitors, and probably a question that's come up a few times, I know, with patients as well during this COVID-19 time. What is the role with BTK inhibitors in the treatment of COVID-19, alongside treatment of COVID-19? And should patients be receiving BTK inhibitors alongside that?
So this is certainly an unknown area, I would say, at this point, but certainly an interesting one. And there's actually some theoretical reasons why BTK inhibitors could be helpful for patients with severe SARS-CoV-2 infection. One of the things that we've learned about the infection is that it's often the inflammatory state that can arise that can cause severe lung issues that can actually lead to patients getting very, very sick. And so the idea is that the BTK inhibitor drugs could, in theory, help to prevent some of that inflammatory response, because some of those same chemicals that get released in the body that cause that inflammatory response are the very ones that BTK inhibitors can block. So there's been some intriguing reports published from the US, including one from our own center at Dana Farber with some patients who had COVID infection and were on BTK inhibitors and did well. And some patients who developed COVID infection and were treated with acalabrutinib in a study led by the National Cancer Institute who did well, but we're talking about very small numbers of patients, and it's certainly enough to inspire larger clinical trials of these approaches. And we now have trials of all the BTK inhibitors open in the US looking at COVID patients, not CLL patients. This is for any person who gets COVID infection and is treated with a BTK inhibitor. But I would caution people against using it without any prospective evidence. I'd really want to see some data from these trials emerging before I'd feel comfortable, because again, we know that these BTK inhibitor drugs can have risks and side effects as well.
Excellent. You've touched on this question a few times, but a patient's asking, should I be chasing the 3 prognostic tests while on watch and wait, or wait until you're needing therapy?
Brian, do you want to take that?
Yeah, I'll take that. I think every patient is going to have a different approach about what they want to know at time of diagnosis. And certainly there's an argument to be made to say, because again, we talk about these test results predict for groups, not for individuals. And both Dr. Davids and I have known patients with all the markers who've gone many, many years and not needed treatment. So, and people with great markers who need treatment in less than a year. So some people prefer not to know, some people prefer to know, but everybody should know before they start treatment. Because if you're 17p deleted, if you have an aberrant TP53, a regime that involves chemoimmunotherapy is off the table. And if you have a complex karyotype in a more aggressive form of CLL, then maybe a clinical trial with a combination is something that you should be talking to your doctor about. So absolutely before treatment, but during watch and wait, I think that's something that you have to discuss with your clinician. But before treatment and also before any subsequent treatment, the IGV IGHV status does not change over time. So you only have to have that done once. But you can not have 17p when you're diagnosed, and it's a very small number to have it at the time of diagnosis. But that number goes up much higher when people relapse, and much more people have the 17p deletion or TP53 who are relapsed and refractory. So it has to be tested not only before the first treatment, but before any subsequent treatments.
Yeah, I certainly agree with that. I want to bring Jason in for a minute here and get your opinion about what Brian was saying about, if you have 17p deletion or TP53 mutation, you absolutely should not be receiving chemoimmunotherapy. But my understanding is in Australia, even in that scenario, you would have to start with chemoimmunotherapy. So how do you handle that situation in Australia?
No, that's correct. So certainly under the standard arrangements for access to medications, we have to use chemoimmunotherapy. That being said, Janssen, who markets ibrutinib, do have an access program where for patients who have deletion of 17p, we can access ibrutinib frontline. So the company actually pays for that, so that's fully funded. And again, they've made an application to our reimbursement agency to try to get a 17p deleted CLL. The problem, though, again, is the TP53 mutations. I suspect there's quite a number of patients out there who don't actually get mutation testing done. So we know about the deletions, but not the mutations. And we know that they carry very similar prognostic implications.
Just thought I'll put one of the questions up early for our participants about who actually has had a FISH test before they've been treated. So Kelsey's going to pop that up for us. And this is the percentage of people on our participating today have actually had a FISH test. So 30% is not sure, and that's not uncommon, I don't think. Yes, 36%, and no, 33%. So we're still very mixed in Australia. What would be the percentage of patients in America where pretty much this is standard, everyone would have a FISH test before treating?
No, so you would think based on what we're saying that everyone's getting FISH testing, and certainly that's probably going to be true at most of the big academic centers, CLL specialists, but the vast majority of patients in the US who are treated for CLL are treated in community oncology practices where practitioners may only be seeing a handful of CLL patients in a year. And so there's a registry that's looked at this question, the Connect CLL registry, and I think in that data set about a quarter of patients were tested for FISH, and even less for IGHV, 10% or so. So numbers don't look too different in the US. I think, Brian, you've looked at this question as well.
Right, and Dr. Mato, I showed the video on, has done a lot of this research, and the numbers are even more dismal in relapsed and refractory patients, where it's even more important to do. So if you're seeing Dr. Mato, if you're seeing Dr. Butler, if you're seeing one of the top CLL specialists in any country, you're going to get the appropriate testing. But in the community, that's why we talk about being your own advocate and pushing to make sure, smart patients get smart care, and ask to try to get this testing done, and ask to have it repeated if you relapse, to have the appropriate testing redone. That we see as the low-hanging fruit. We can make such a different impact because we have such great treatments that are available, but patients aren't always getting access to them because they're not getting appropriate testing done in advance.
And would this be reflected with your practice, Jason, in Australia, you feel?
No, look, I'd be surprised that the number of tests were that low. I suspect that, again, it may be a lack of communication from doctor to patient about those particular results, rather than the test actually not being done. Most CLL patients will be looked after in fairly large public facilities, so certainly accessibility of the test would be fairly high. So I'd be surprised if they weren't done more often, but I think it's more of a communication failure rather than actually not doing the test.
Interesting. It's something for patients to go back to your doctor and ask whether you've had the test. Sorry, Deb.
Yeah, I would say that I paid, I think it was about $2,000 in 2012 for the IGHV testing, and that was because of that wonderful website and Chaya Venkash on CLL topics, who I was, reading back in 2012 and found out the importance of IGHV testing. And the reason I wanted to know, even though actually it wasn't, I was still going to have FCR. It still wasn't going to change what they could do for me in Australia, but mentally I wanted to know how much I needed to prepare myself to throw the kitchen sink at my disease. And if I'd found out I was mutated, I would have been much happier. I remember being devastated, and just in case my specialist at Cabrini Hospital wasn't aware, which she was, they actually, when they sent the pathology letter, they said, this is a poor prognostic indicator for CLL. And from that point, it was much easier for me to prepare myself mentally that, yes, I was going to have a transplant, or I was going to have aggressive treatment, that this wasn't something I was going to be able to live with for years, which if you're mutated, you really, I know patients, as you will, Brian, that they haven't needed treatment for 20 years. And sometimes when, people contact me, I have to say, please don't Google, please don't worry too much, because a lot of people are not going to have treatment for this. And if you know you're mutated, I think it does give you a bit more peace of mind. So it's whether you want to know, and unfortunately you may still have to pay for that test, but it doesn't change. It's one of the few things with CLL that doesn't change. Yeah. Our cytogenetics change substantially over the course, but whether you're mutated or unmutated, and it just, for me, I needed to know. So I would have done it early in Watch and Wait, and I did.
And that leads into the fourth poll that, Kelsey, you can pop that up. So we ask patients if they know they've had an IGHV test. And as you mentioned before, this doesn't change. So this can be done at diagnosis, correct? And so at least you know what your mutation status is. So again, most people aren't sure, and I think that's probably what Jason was mentioning before. A lot of doctors will probably do this test, and you may not realise in a lot of public hospitals in Australia, and only about 9% actually know if they've had this test, and 20%, almost 20%, definitely don't think they have either. So I think it's a bit of, again, a communication thing there too, but ask your doctor when you see them. Thank you.
Donna, I wouldn't be surprised if that data is a little bit more accurate than
okay.
Yeah, I suspect, in fact, the understanding about the prognostic implications of that test probably hasn't percolated its way into a normal general haematology space. So if you work in a large centre like the Peter Mac, for example, some of the other larger centres, it probably is done. But yeah, no.
It's done routinely at Peter Mac, that's for sure. Yeah.
One big word that's really important to understand in CLL is heterogeneity. So Deb, Deb mentioned this, but it's worth knowing. You're hearing about all of this stuff. You hear Deb's story and my story, but we're one end of the spectrum. And Dr. Davids will tell you about anywhere from 15 to up to maybe 30% of patients who never need treatment, who have a normal life expectancy. So for me, I'd want to know that as best I can. I want to know which group I'm in. I know that it's not going to predict for me, but I want to know what are my odds, like Deb wanted to know. But I don't want, you tend to get the patients in these kinds of webinars who are the ones who've been through a lot to tell their stories. But the patient who's had a boring watch and wait for 10 years isn't going to show up on one of these webinars. But Dr. Davids has those patients. Dr. Butler has those patients they see once a year. Yeah, your blood count's fine. See you in a year. Goodbye. And God willing, most of the people tuning into this are in that group. But we know, more than a quarter of patients have that story.
Excellent. Just a question that came up, just to clarify again, does deletion 13q indicate a more or less favourable outcome?
So yeah, deletion 13q is both the most common cytogenetic abnormality, about 55% of patients at diagnosis, and it's also the most favorable, particularly when it occurs as a sole abnormality, meaning that's the only thing we see on the FISH test is the 13q, and that's a very favorable prognosis. Although it can be mitigated a bit by unmutated IGHV, so then you sort of get into this mixed prognostic picture. You can also sometimes see 13q with other abnormalities like 11q, and that's probably a little more aggressive, but yeah, when we see 13q alone, particularly if it's mutated IGHV, quite a few of those patients are the ones who are going to go decades without needing treatment.
Excuse me. And there's another question for a couple of patients. They've been diagnosed with SLL and early, so early 30s. Does it suggest usually that the younger the age, the quicker the disease progression for these patients?
So not necessarily. So there have been some studies looking at this, and really the most important factor is the prognostic factors. The genetic tests we've been talking about: FISH, IGHV, and TP53 status. Regardless of the age of the patient. I think the challenge with young ones like Deb, when she was diagnosed in her 30s, is that you have this long time horizon. As the CLL doctor, your goal, rather than your 80-year-old who you're trying to get 5 to 10 more years of life, now you're talking about a 38-year-old and you want to get 50 or 60 more years of life. I mean, it's a more daunting task because I think, as Deb was saying, the sequencing and how you approach this, it's really more of a marathon than a sprint there, and you have to plan out everything and think ahead as to next steps. Becomes really important in that respect. But strictly in terms of prognosis, I encourage people to think more about the genetic prognostic markers rather than the age of the patient.
Excellent. I'm just going by the votes. There's quite a few votes for this question. What are the current research findings regarding familial CLL? And are there any repercussions for patients with existing family links?
So there's clearly a genetic component to the development of CLL that's been established by a number of different studies. The challenge is that we haven't really identified the causative genes. In other words, there's no genetic test that we can send to really look for predisposition to CLL. We certainly see families where CLL runs in the family, and we know there's clearly a risk there. We have sort of indirect evidence from, for example, the fact that in East Asian populations, CLL is much less common. And when the East Asian population, when people move to the United States, for example, that risk doesn't tend to change all that much. They're still at a relatively low risk. So like with most things, there's probably some environmental and familial component. What I counsel my patients is that just by virtue of having CLL, that their first degree relatives, their siblings, and to some degree their children are at an increased risk of developing CLL someday. But that being said, CLL is still a fairly rare disease. And so the likelihood that their family members will develop it is also low, but it is something they should be aware of.
And I would add that, you know, the absolute increase might look bad, like double, but if the risk is 1 in 10,000 and it goes up to 2 in 10,000, it's still a very low risk, even though it's double the risk. Also, CLL patients, their family members have a higher risk of other lymphomas and blood cancers too. So it's not just CLL, but the risks are still absolutely quite small.
Brilliant. And another common question, especially in this COVID-19 time, a lot of people are worried about their immune systems and the restoration post-treatments for their immune systems. What are the prospects of restoring a normal immune function in patients who are in remission after they've had, say, FCR or other novel therapies?
So this is a really challenging question to figure out for any individual patient because there's a lot of different factors that go into the immune function, and our measures of this are relatively crude. So for example, the example here of a patient who's had FCR, there's kind of two main things going on. So one is that the CLL hopefully has been put into remission. So some of those immunosuppressive effects of CLL are hopefully less at that point. But on the flip side, the FCR, particularly the fludarabine, can have very long-lasting effects on the functioning of, for example, the T cells, which help you fight infections. So you kind of have two opposing forces there, and it's hard to know in any individual patient which one's going to win out. But I think in general, we have a good sense that CLL patients do have some compromise of the immune system throughout their course. The patients who have lived with CLL for longer, and particularly those who've been through multiple rounds of chemoimmunotherapy, seem to be at the highest risk. Patients who are relatively recently diagnosed do still have some compromise of the immune system, but perhaps not as much. But I think all CLL patients do need to be quite careful around the COVID-19. We've had some initial reports now of the outcomes for CLL patients, and not surprisingly, patients have had a tough time with COVID-19. A lot of that is because CLL patients tend to be older, and being older in general is a high risk for the virus. But there probably is some additive risk of having the CLL in terms of the immune suppression.
Excellent. And a question that keeps coming up quite a lot as well, do you recommend having flu vaccinations, maybe a booster, having one out 3 months apart sort of things for like a booster during the flu seasons?
So I've never seen any evidence supporting multiple flu vaccinations for CLL patients. We certainly recommend that all our patients get a single vaccine. There's some weak data suggesting perhaps the higher dose flu vaccine may be a bit better, but I'm not super convinced on that. So I think it's just important to get some kind of flu vaccination on a yearly basis. I don't know if Jason, if you've heard of a sort of double vaccination strategy for flu, but.
Not specifically for CLL, we certainly, there's a little bit of data, broadly speaking, immunosuppressed patients that a second dose perhaps can improve the quality of the immune response. But as you were alluding to, it's quite low level data. In Australia, the second dose would have to be self-funded as well. So the National Immunisation Scheme will fund a single dose, but not the second one. But it's a reasonably small expense. It doesn't seem to increase toxicity.
Okay.
And I would add that it's really important for CLL patients when newly diagnosed to talk to their doctors about their immunizations, because generally your immune system doesn't get better over time with CLL. So getting your pneumonia vaccines and catching up on any vaccinations that you might need that are age appropriate, gender appropriate, is a really good idea to do early on in the course of the disease. Make sure your boosters, make sure all of those things are up to date. This is a big part of what you do in that active observation or watch and wait period, is get yourself up to date. And getting the flu jabs, I think, is something that's critically important. What we haven't been able to do, and what I'm urging Dr. Butler and Dr. Davis and everybody to do, is find a way to reconstitute our immune system. You've knocked back the CLL, you've given us a longer life, but we still remain immune suppressed, most of us, even when our disease is knocked way back. and we still have a higher risk of problems from infections, not just COVID, but all infections. So that's one of the big unmet needs in CLL.
Yeah. And I'd just add to that you've got to make sure you're keeping up with all your checks. Even though I'm in the middle of a CAR-T trial, I've got a new GP, and the new GP said, right, Deb, we need to sort out your smear test, your skin check. It's the risk of secondary cancers is much higher for us. So just make sure all the things that you should be doing as standard looking after yourself, you're doing. And that's incredibly important, particularly at the moment when none of us, I'm in Melbourne, haven't left my home. So it's really important that we're looking after our general health as well.
Exactly. Very good point, Deb.
Yeah, Deb, I'll just reinforce particularly the bit about the skin checks. We know that patients with CLL have very high rates of skin cancer disease, mainly sort of basal cell carcinomas and squamous cell carcinomas, but probably melanoma, and that significantly interacts with the sun exposure that we all have in Australia anyway. So please maintain very regular skin checks. It's critically important. That in itself can save lives as well.
Exactly, just keeping as healthy as possible. Excellent. We probably should have mentioned this before, but what is involved in having a FISH test or a blood test or bone marrow biopsy?
Well, it's a FISH test from the blood is a pretty straightforward procedure for the patient. It's just like any other blood draw. It's a complicated procedure in the laboratory that takes a pretty sophisticated lab to be able to tease out those different chromosomes with all the different probes and so forth. The second part of the question, I think, was about a bone marrow biopsy. That's a more involved procedure and more invasive, obviously, but it is done typically in the office setting with some local anesthesia, and it can be a bit painful. I haven't had one myself, so Brian can speak to that, I'm sure, Deb, better than I can. But it's something that our patients can get through, and certainly gives us very valuable information.
So bone marrow biopsy, as Dr. Davids pointed out, is not routinely needed for the diagnosis, and there's only very specific times it's needed in the management of CLL. I've probably had more than 10 with the transplant. I'm not going to say they're pleasant, but I'd prefer to have a bone marrow biopsy than see the dentist. I mean, the local does a good job. It works for me. Other people have a much more difficult time with it, and even some places they do conscious sedation to knock them out. So everybody's variable how sensitive their bones are, but I've never found it. I've gone hiking an hour later after having the bone marrow biopsy done and never needed anything more than paracetamol, acetaminophen for the pain. It's never been a big deal for me.
I've had 23
Wow.
and they've told me I'm going to be having them very regularly over the next few months. I have just discovered, and this is a uniquely Australian thing, so ask for the green whistle. I was only told about this 3 bone marrows ago, and I'm angry that I'd had conscious sedation and nothing when I was in the NHS system. They don't even give you anything. Green whistle, people in Australia. It's Penthrax for the doctors in America. And it I've never done drugs in my life, but had you ever done drugs, I'm told it equates to a hit of something and then a nice sort of feeling as if you're 3 or 4 joints down. And then 2 hours later you can drive. And I have to say, I will volunteer to have a bone marrow to get that green whistle.
And being a nurse, I've actually had to tear it away from patients. Yes, it's what they quite often use on ambulances, actually, for people in severe pain. So, yeah. Okay. What is the current success rate or prognosis for people having bone marrow transplants? Or allogeneic transplants, I should say.
So that has improved over the years. We've looked at least at our own center. Over the last 5 years or so, we haven't done very many, but sort of the decade or so before that, the numbers continue to improve. Not necessarily so much because the chemotherapy got better, but I think the supportive care got better. We got better at treating infections and figuring out how to treat graft versus host disease, where the donor cells could attack the body. And so, if you look sort of in the long-term series, it ends up being at about 40% or so of patients who remain progression-free long-term, 5 or 6 years out from the bone marrow transplant. So many of those patients probably are cured. But unfortunately, still probably about half of those patients have pretty significant graft versus host disease. So even if they're living without the CLL, they have other issues to contend with. So it's something we try to avoid if we can, but when we need it, can be potentially curative for some patients.
Excellent. This is a question for Deb, perhaps. Given our situation in Australia and the need for advocacy and political pressure, I'm assuming with the having testings and different drugs being introduced, what is the best way for us to develop stronger means of influence? So being an advocate for this.
Yeah, absolutely. Lymphoma Australia is by far the most, I've found Sharon's connections in Canberra, the lobbying that Sharon personally does. If you're interested in helping with advocacy, please reach out to us here at Lymphoma Australia, because more voices do make a difference. And it's a shame that it had to take me being in newspapers and on TV and jumping up and down and losing my privacy. I have to say, finding other patients willing to be public is really important. There was one point where the Adelaide Advertiser, I've never even been to South Australia, wanted to feature a patient, and I couldn't find another patient locally who was willing to tell their story. And I think if we're willing to give up a bit of privacy, it helps make politicians realise how important this is, because stories make a difference. And hearing bad stories, so there's cases I know where people didn't have the great outcome I had because they couldn't get venetoclax, and that's tragic. And their children are growing up without their parent because they couldn't access it and I could. So I feel very strongly. We're very lucky that with clinical trials here. Obviously, we're a publicly funded system, so we do have to justify the cost of these drugs. But tell your story and reach out to Lymphoma Australia, because we'll help share your story if you're willing to help us. It's very important that we speak out, particularly about the testing.
Exactly.
A lot of CLL patients keep proud about it. And I think all of us know some really quite famous CLL patients who could make an impact and make a difference, but they keep it like a closet disease because they're afraid it'll affect their career, promotions, other things like that. And if we could get people to be more public about it and talk about, because it's the poster child. It's easy, the poster child for cancer research, because you can get a biopsy done every 3 months because you just have a blood draw, and that's a biopsy of the CLL. There's so much great research going on in CLL. So if people were more out there and more open and more involved in clinical trials, things would be moving even faster in the CLL world.
You're saving lives. Brian knows himself. Brian's probably been instrumental in helping save my life by what he's done with the CLL Society. If he hadn't run that conference, I wouldn't have been in California, and I wouldn't have met John Gribb, and I wouldn't have got on a clinical trial. I'm on the CAR-T trial because I sat next to Michael Dickinson on a plane to Canberra. I mean, it's random, and it shouldn't be so random. But I worked in politics for a long time, and the politicians didn't even know I was sick. Greg Hunt, the health minister, I decided not to be public until I had to be, because I'd had to move overseas to access a drug that had been basically invented in Australia and couldn't access it here. But yeah, if you're willing to help, we'd be very grateful to have your help. I'd like to become private.
I just thought I'd put up another poll that we had throughout the presentations as well. So it leads into the question about your own self-advocacy and also advocacy for new drugs coming into Australia. We asked whether people have actually been asked or feel they've had enough information about clinical trials or been asked to be on a clinical trial. And there's an overwhelmingly 62% said they do not know enough about clinical trials in Australia. So I guess organisations such as Lymphoma Australia are always advocating for these new drugs, and a lot of these new drugs are on clinical trials. So being an advocate for yourself and asking your doctor what is available to you or in your area, or if you wanted to travel to interstate it's a bit difficult at the moment, but looking at different options and being an advocate, such as what Deb and Brian have both been for themselves. If you want to comment on either of those, both of you?
Well, I would just say, and I think Deb would echo this, I would not, if I'd have followed conventional therapy, I wouldn't be alive today. And there's never a good time to have CLL, but there's never been a better time to have CLL. And what's available now is just incredible. But I was in 2 phase 1 trials. I mean, and I would not be alive if it wasn't for those 2 phase 1 trials. And I think Deb would say the same. So I'm very keen on, and Australia has a lot of hot trials going on, but you've got to sometimes be your own advocate, because your community hematologist may not know about them if you're not at a big center, if you're not at a big teaching facility. So you've got to really push and look, and it can be, there are places to look online for trials, clinicaltrials.gov, but it's not easy and patient-friendly. Lymphoma Australia, you can help research and help you with that kind of guidance and stuff too. But people have this vision sometimes that you're a guinea pig. No, you get the best possible care. And there's no, they don't give you a placebo that does nothing. They give you, usually it's the standard of care, the best possible treatments available versus the new treatment. That's usually the way these trials work. The only time they give you a placebo is when there are no treatments that are known to help. It's very rare that these things, so you're not a guinea pig, and you can ask and you can look into it and decide that you want to get out of it later. Or you don't, once you've committed, you can leave 5 minutes before the trial's ready to start, so, but like I say, trials are life-saving, and the docs who do them are often the absolute top docs. You get very close supervision. People are monitoring you carefully. I can't recommend trials highly enough.
Yeah, I'm the same. I'm conscious of time, so I'm going to hand over to Butler to give his impression, because I feel as strongly as Brian. And the phase 1 trial that I'm about to do, it's, this is real first in human stuff. The last phase 1 trial I did was a combination therapy, and both drugs were in phase 3 trials. So I was much more confident about that than I am about the next one. But hey, fortune favours the brave, I'm hoping. Dr. Butler, I'll let hand you to give your impression.
Yeah, look, I'll just say that I think if you reach a point in your disease status where you need treatment, one of the questions you should ask is what trials are available. And keep in mind that each of the individual centres in your city or close to you could have a very different clinical trial portfolio. So if your centre doesn't have that trial, then cross referral to another centre in your city can be a very convenient way of getting access to a really good trial. Under normal circumstances, interstate travel, for example, to go to the Peter Mac would be a very good option as well. But clearly with the closure of borders at the moment and the need for quarantining if you move interstate can be very challenging. So it's perhaps something that's changed a little bit. And I don't think we've quite yet embraced the virtual telehealth arrangements for clinical trials. To an extent we have, but it's still quite limiting because the need to deliver actual treatment that's part of the clinical trial. But yeah, a very important question to always ask if somebody's recommending you need to start on treatment is what trial options you have. And you might be surprised at the number of options that are available.
And it leads to Dr. Davids. How many of your patients would you be putting automatically on a trial at time of treatment?
So if you look across the US at cancer clinical trials, I think the estimates are somewhere between 5 and 10% of patients are treated in the context of a clinical trial. This is across all cancers. And I would say of my patients with CLL who are receiving treatment, it's probably 85 to 90%. So we try to get every patient to consider a trial. Trial's not always right for every patient, so certainly respect the decision of patients who choose not to go on trials. But I tend to be pretty persuasive. You can see with those numbers. So most of my patients wind up on trials.
Yeah, that's excellent. And I guess that's back to you going to a specialist centre to be able to get the information and the treatment that you need as well. Look, I know there's a few more questions there, but I might need to considering the time and needing to, I might answer these offline for those patients. I can answer some of those ones. But I just thought I'd just finish off with the last poll, just to show everybody that there are lots of people across Australia and internationally on today's broadcast. Thanks, Kelsey. Maybe not. Okay. It's all right. We're basically, everyone, here we come, every state and territory. So, and thank you to all those people from overseas as well. I'm assuming from the States have joined us today as well. So just sort of show that you aren't alone on the screen. It's can be quite difficult in a webinar to sort of know that there are lots and lots of people on the other end. We're not just talking to ourselves. So it's great. And thank you so much for everyone for joining us. And particularly, Jason, do you want to close off, or Sharon? There's some great questions there, and I will get to those people.
Thank you, everyone. Yeah, just on behalf of Lymphoma Australia, thank you so much for joining us today, and especially this wonderful panel. This has probably been the first time where I've seen such an honest and open and frank discussion around the disease, and I truly wish that something like this had been available 20 years ago when my mother was diagnosed with CLL. But as Brian said, there's never been a better time as well to have maybe a cancer like CLL because of the advances that have been made. And the purpose of this with Lymphoma Australia is it is about giving everybody all of the information so that you can actually then make the best decision and be the best advocate for yourself. This isn't about what is going to work for all patients, but what is it going to work for you? And we want to be able to provide that information. We feel very privileged to have had Professor Davids as well. Thank you so much for just giving us all of that information so that patients now can actually take it back to their own doctors, and hopefully we can actually move forward for every patient, because like we said at the start, you become your own advocate, but you're also advocating for everybody else. So the more people that we can actually help along the journey to actually have the same outcome, which is the best outcome for them, is just what we want to be able to do. So we really appreciate the time that you've given us from America. Unfortunately, we can't give you our Kubra hat, thank you, and everything else, because we can't have you here. But when we get rid of this pandemic, it will be wonderful to meet you in person, and thank you from everybody as well. And Brian, I think you're just in a league of your own when it comes to patient advocacy. Myself, like Deb, we met overseas. We've had you over here in Australia. The work that you're doing for every patient, we cannot thank you enough for as well. So thank you as well so much for joining us from America. And Deb as well. It's patients like yourself that are actually helping us help other patients every step of the way. So we hope that we're going to be able to continue to have these. We don't want, we like the boring patients and the challenging patients like our Brians and Debs, because you do actually bring us the whole package, so that it's like, well, it's great if I'm a boring patient, but those challenging patients, you really help us join the dots. And thank you so much, Dr. Butler, for bringing us as well the Australian perspective, because we've got a lot to do here in Australia. We're not up to the America or potentially Europe yet, but by Dr. Davis and Dr. Kaufman sharing with us, what we can actually do is work together to get all of our voices together so that we can actually bring all of these treatments to Australia and potentially other places of the world that are also lagging behind with us. And then just in closing, I would also like to mention, yes, clinical trials, such a big thing, something that as a country we need to do better in. We need to have more patients going to clinical trials. But for CLL patients, I would like to especially mention, have a look at the ALLG website. There are clinical trials there. You can also download the ClinTrial Refer app. Again, this will actually show you which clinical trials are available in Australia, but contact Lymphoma Australia too, because we'll actually help you unpack that. We don't want COVID to be a reason either for you not to get on a clinical trial. If we have to cross a border, we'll get across a border if we need to. Thank you so much, Donna, as well. And also, thank you so much, Janssen, for helping us package this together. Again, a pandemic is not going to stop us from getting out to everybody. We've worked out technology. We've had a few glitches, but otherwise we've managed, so thank you to Streams. But again, thank you to our international guests. Thank you for everyone for joining us here today. We truly appreciate it.
Thank you.
Thank you.
Thanks so much.
Bye.
Bye.
Bye. Cheers.
Thanks.